Greetings, everybody. I'm Shreya. Today, me and my teammates are going to present our project on the topic microbial production of carbohydrate-based polymer under the guidance of Dr. Vishwambar Mishra. So the main aim of our project is to produce carbohydrate biopolymers from a new potential microbial strain. And the objectives include first to identify the microorganisms that has the capacity to produce carbohydrate biopolymer, and then to identify the produced biopolymer through characterization assessment, and then the optimization of the produced carbohydrate biopolymer from the isolated microbial strains. And the novelty of the project is to produce a carbohydrate biopolymer from a new microbial strain. And this novel strain may possess unique characteristics, unique metabolic pathways, etc. And these microbial polymers have various applications in various industries and are also environmental friendly. So microbial production of carbohydrate biopolymers is basically a process where we produce biopolymers from the microorganisms such as bacteria and fungi, and exopolysaccharides, which are also known as EPS, are the polymers which have high molecular weight, and these are mostly composed of sugar residues and are secreted by microorganisms into the surrounding environment. And these exopolysaccharides mostly consist of monosaccharides and some non-carbohydrate substances as well. And these have again various applications in various industries, but especially in food and pharmaceutical industries like pullulan, gelin, xanthan gum, leaven, etc. And benefits of using these biopolymers produced from microbes uh, are like they are biodegradable and biocompatible, and these can be customized to have specific properties like flexibility, strength, etc. And these can be produced from renewable resources as well, such as biomass. So coming to the literature review, this paper talks about the production of biopolymer called pullulan from a novel microbial strain called Micrococcus luteus. And after the characterization, it revealed the presence of alpha 1 to 6 and alpha 1 to 4 linkages in its structure through FTIR and enzymatic hydrolysis analysis. And the optimal parameters were determined as temperature 35, pH 6, and RPM at 250, and an incubation time of 5 days. And through the characterization, it was also found out that it has peptone has a significant role in the production of this biopolymer pullulan. This paper also talks about the production of the pullulan biopolymer, but from the different microbial source, that is culinary leaf samples. Here, among 100 isolated strains that they had, only one strain could produce significant amount of pullulan, and that strain was identified as Aspergillus japonicus. And among other microbial strain, this microbial strain has potential for commercial use in the pullulan production. And this paper is talks about the production of EPS from a microbial strain called Bacillus pumilus, and here the microbial source is water irrigated soil. And this EPS is a heteropolymer, and it composed of mannose, xylose, glucose, etc. And this EPS specially has high molecular weight, that is of 218 kilodalton, and thermal stability up to 180 degrees Celsius.
These are the materials that we are using in our project, and the methods that we are using are serialization, streaking, fermentation, microfiltration, and etc. The process flow include: first we screened the microorganism, and then EPS was produced from the isolated strain. Then we isolated identification the potential strain. Later characterization and optimization were performed. Coming to the procedure, first first we did the serial dilution by taking moisture sample from college campus as a microbial source, and then we prepared the three agar plates, and then serial diluted solution was spreaded on the agar plates that we prepared. After that, incubating agar plates overnight, colonies were produced. Next. One colony from each plate were visualized under the microscope. Designation SM-A and SM-B and SM-C were given to each stain. When these stains were visualized under the microscope, it was found out of the first stain SM-A was cocci-gram positive, and other two stains SM-B and SM-C were cocci-gram negative. These stains were again individually sticked on the new petri plates to get pure culture. For the growth medium, we prepared 3 flasks of 100 ml fermentation media according to the composition given here. The, the three stain SMA, SMB, SMC were incubated to the 3 flasks respectively and fermented 15 days approximately on the orbital shaker. Later, later 5 ml of each floss were taken and centrifuged at 7,000 rpm for 15 minutes. Plate was discarded. Pellet was discarded and supernatant was precipitated using cold ethanol. And then wet and dry weights were noted down every alternative days.
At the commencement of our project part 2, we found out that the fermentation media were over fermented, and so we prepared new fermentation broth and repeated all the prior steps. After 15 days of incubations, we have collected the wet and dry weights of the samples, and from the calculations, strain SMC produced more wet and dry weights than the strain SMA and strain SMB. Then we submitted these strains for FTIR analysis, and these are the spectra. This is a spectra of sample 1, 2, and 3. From the analysis of the FTR spectra, we found out that sample 1 has simpler molecules due to low bending, and sample 2 and 3 have complex molecules due to presence of bending and stretching. These complex molecules are similar to polysaccharides. We later next. We later submitted a strain SMC for molecular identification, that is for 16S rRNA sequencing, as it produced a great amount of dry weights than other strains, and it has complex molecules. This is the obtained 16S sequence. When we ran the sequence on NBLAST, we found out that the organism was Staphylococcus epidermidis. Along with our guides, we have submitted the sequence on GenBank. Then we later conducted optimization of parameters of time, pH and RPM, so as to produce a significant amount of EPS. When it comes to optimization of time, we have taken the time in days, and we have noted that on 4th day, the EPS production peaked, that is, it produced 25 mg per ml, and later it decreased significantly. For pH, we have found out that the EPS production peaked at slightly acidic conditions, that at pH 6, and at neutral and basic conditions, the EPS production decreased significantly. When it comes to RPM, we found out that the best RPM for production of more EPS is at 200 RPM. At 250, the EPS production steep down. We conclude that the strain SMC produced more EPS than the other two strains, and we identified the organisms to be Staphylococcus epidermidis. With FTIR analysis, we identified the produced EPS to be polybeta 1,6-N-acetyl glucosamine, which is also known as polysaccharide intercellular adenosine, which is involved in which forms a biofilm around the bacterium at extreme conditions. Uh, to obtain significant amount of EPS, we optimize the conditions to be slightly acidic, that is at pH 6 and at RPM of 200. In conclusion, biopolymers are important because they're natural materials and they can be used in many ways, like making eco-friendly packaging and biomedical products. They're good for the environment because they come from renewable sources and are biodegradable. Using biopolymers help us protect the planet and make things better for the future. These are the references we have taken for our project. Thank you.